Namaste and thank you very much for joining today's KSAS lecture series. We have a prominent speaker today, Dr. Neeti Sanan-Mishra ma'am, who is the group leader at the International Center of Genetic Engineering and Biotechnology, ICGEB in New Delhi, India. She has around 30 years of experience in research and teaching. Dr. Neeti leads a dynamic team engaged in groundbreaking research projects on the use of RNAi technology to address passing climate change associated challenges in agriculture. She has published numerous research articles and received several awards and patents in her credit. So we welcome you, ma'am. Today she is going to talk on the topic role of NGS in analyzing the micro-RNA-target interaction in rice. So I am from the philosophy background, so I have not deep understanding about it. But as per my surface understanding, the next generation sequences that plays a crucial role in studying microRNA target interaction in rice. As we are from social science and you are from science, so we have some responsibility for society. So my understanding, this can target the interaction in the rice using next generation sequencing had the significant role in societal benefits. It can help improve the rice variety by enhancing stress resistance, disease tolerance, and adapt the better to climate change. So additionally, this research, if these outcomes will come in the society, this research can support the global food security by developing the rice varieties that can thrive in challenging environment, uh, ensuring stable production for growth population. And this is the challenge of this time, especially in India, to help the primary sector of the economy, primary sector of the society, which is agriculture. So we welcome you, ma'am, for your talk. Over to you.
Well, thank you very much. And greetings from my side and to people within India and outside India who are in different time zones. So, and at the onset, I would like to thank the organizers at KSAS for, finding me, my research interesting enough to have me here. As was said, we as a group are looking at RNAi processes in plants. Our focus is on microRNAs, trying to understand how are they regulating the different aspects of plant development. And for today's lecture, I have chosen a very basic topic, which is more of a methodology oriented or a basic computational biology oriented talk on how we are using the NGS analysis to fish for genes that can be then selected for downstream functional analysis in a search for loci that can be edited or manipulated so as to improve the response of rice plant to stress. Because we all know that we are living in an area of, or in an era of global warming, and rice plant per se is very sensitive to stresses at the early seedling stage and at the heading stage. And these stresses impact the yield of rice. So without getting into the relevance of climate change and production and rice, I'll try to keep my talk focused on NGS and how it has helped us in sifting through the data to finally get hold of the genes that may be playing crucial role in modulating the physiology of the plant. And when some of these we have now selected and are taken forward for functional analysis and also for now trait manipulation in the rice plant. So to start with, I thought we can start with the central dogma, because this is textbook knowledge, and people with even school level or basic level of science understand this, that how the central dogma evolved. If we go back and look at science, it moved from one gene, one protein, to one gene coding for one polypeptide, making a protein. Then the concept came of one gene coding for multiple polypeptides. The concept of splicing came in. And finally, we know that in the DNA, there are multiple genes, and these genes perform the functions. The link between DNA, which is the storehouse of the genetic information, and the proteins, or which are the enzymes and are considered to be the workforce, is the RNA molecule. And RNA for long has been recognized as the messenger, because it was thought that the basic the function of the RNA was to transmit or translate the information coded into the DNA to form functional proteins. However, this concept has now changed, and we know that RNA is doing a much more important role in the plant, including regulating the translation, that is a coding of information from RNA to proteins, as well as transcription, that how much of the DNA will be coded into the RNA. And all this is being done by a large family of non-coding RNAs. So there, for the sake of RNA silencing, the classical molecules that we talk about are so-called the SNC RNAs, or the small non-coding RNAs. They also constitute a big family, which have been classified as per our understanding into different categories like siRNA, micro-RNA, tRNA, so on and so forth. But most of these, or rather all of these, are involved in regulating development as well as environmental adaptations in plants. Also in animals, because the RNA silencing process is conserved in the eukaryotic cells. And these small non-coding RNAs modulate the gene regulation at the epigenetic, through epigenetic silencing, that is at the DNA level, at the transcript, by transcript cleavage at the translational level, and by repressing translation again at the transcriptional level. So different modes by which they can regulate how much of DNA will be converted into RNA, and whether this RNA will be available to make the protein or not. So this can be summarized as saying that they are a part of a complex adaptive system which has evolved to monitor and modify the gene expression of the organism so that it can respond to the environment. And the rules are so far poorly understood or poorly defined. Classically, if we look at literature, the response of RNAi was first noted in 1990 by a group of georgesons in a flowering plant called petunia. Now, petunia normally is a dark, bright colored flowers which come during the springtime. This is the time when these are flowering. And darker color flowers or brighter color flowers are preferred by people. So we all know that color development is regulated by the CHS, or the chalcoin synthase gene. So based on the classical central dogma where DNA makes RNA and RNA makes protein, the hypothesis was that if you make more RNA, then you will have more color formation. So the color of the flower should be more darker. With this concept, they took the CHS gene and they tried to overexpress it in the flowers, thinking that more enzyme will catalyze more color formation. But what the product, when they looked at the product, everybody was amazed, because the flowers that resulted were either white, indicating absence of color, or they had patchy experience. So something or some mechanism was triggered which caused this switching off of gene expression rather than the switching on of extra gene expression, which was poorly understood. But over the period of time, a lot of research was done, and people did understand the concept of silencing, where and this was termed as initially termed as a phenomena of co-suppression in plants, where when you try to overexpress an endogenous gene, some mechanism gets triggered which suppresses its expression. And now we have, over the years, a lot of work was done, classic work done by the Fire and Mellows group, who recognized or who identified these small RNAs. And now we know that there are two distinct processes occurring in the plants. One is gene expression, where the protein is formed, and the other mechanism is silencing, which is facilitated by these so-called small RNAs. They are at the and these will go off and cleave or shut off translation, so that protein is not involved or not formed. And the overall response of the cell is actually a balance between this expression and silencing. So this flower represents the dark red color is a classical case of everything, every gene switched on. So this is gene expression. And the white flowers indicate silencing or switching off of gene expression. But this is not all. Nature has a method of regulating spatial and temporally how and when a gene will be switched on or switched off, and to what degree. So we can actually have varying gradients of expression, resulting in a diversity which can be seen in nature, as indicated in this slide, giving us beautiful patterns, color gradients, and as well as different color formations. So what we need to understand is not expression alone, neither silencing alone, but how a balance between the two is established to get a functional gene product. And all this, rather a major part of this, especially the silencing part, is brought about by a large family of RNAs, which are called the non-coding RNAs, because majority of them will not code to form functional proteins and have regulatory functions. These include the long non-coding RNAs and the small non-coding RNAs. And our group is more fascinated by a small subgroup of these small non-coding RNAs, known as the micro-RNAs. The interest lies because micro-RNAs originate endogenously from genes from within the genome. There are specific genes which will code for large transcripts called the pri-mRNAs. These are highly folded structures and are processed sequentially by a set of enzymes which form the microprocessor complex. And in this DCL, or the dicer-like one, is the major cutter or the dicer. And this will gradually process this long transcript into small RNA duplex, which is about 24 nucleotide long. Once this nucleus is formed, it is transported into the cytoplasm, where it gets associated with another set of proteins to form a functional RNA silencing complex to bring about cleavage or translation repression, so that protein formation will not occur. Now, in plants, this system is highly regulated or very stringent and compartmentalized, where the microRNA biogenesis, that is origin, is compartmentalized or restricted within the nucleus. And once the functional molecule is produced, it is then transported into the cytoplasm to bring about silencing, mainly at the post-transcriptional level. So this makes microRNAs very important and interesting molecules, because these are the small RNAs which will regulate the entire development of an organism. And if we look at the function, then they are regulating almost every aspect of the plant biology, whether it is cell division, organ identity, organ polarity, so on and so forth. They help in maintaining the chromosome or the genomic stability by regulating the transposons. And they also play a major role in governing the stress responses of the plants. And this is becomes very important in the present day concept of climate change. So, and just to, as a quick understanding of how microRNAs act, we know that they can act as at the post-transcriptional level by either cleaving, that is cutting the transcript, or by repressing it, that is stalling the translation so that protein is not formed. And this pathway constitutes the PTGS or the post-transcriptional gene silencing. And some of them may go into the nucleus and they can stop the transcription of the gene by heterochromatinization and other processes. And this is known as TGS or the transcriptional gene silencing. So, microRNAs then, can be linked or can be not linked, can be equated to the transcription factors and are considered as the master molecules in the cell because they are naturally occurring, they can impact one or more genes or pathways, and they can regulate a large number of endogenous genes and pathways. The specific interplay comes when the microRNAs regulate the transcription factors or the transcripts encoding the transcription factors. And these very transcription factors are the ones which in turn regulate the formation of the microRNA because the microRNAs are produced from the endogenous gene. And they form cyclic loops, regulatory loops in the cell in which they not only regulate each other, but they also modulate the battery or the thousands of the downstream target genes regulated by each of them individually or together. And through this, they can bring about a variety of response. So there are specific microRNAs which belong to large family members like 156, which targets SPL. 156 is a big family of microRNAs, which is targeting another big family of the SPL transcription factors. Similarly, 172 targeting AP2, so on and so forth. So complex genetic nodes or regulatory loops exist within the cell, and these need to be understood to understand the dynamism or the gradients which are established within an organism once it grows over a temporal, over the time, and understand how these gradients are spaced spatially, that is within the organism, within the plant, as well as temporally during its growth cycle to regulate the overall phenotype of the plant. Just for explanation, and this chart is not to scale, this knowledge has been derived from studies done by different groups in Arabidopsis. So in Arabidopsis, we know that there is one transcription factor called SPL, which regulates the vegetative phase formation. So as the plant grows, the SPL levels accumulate, and this would regulate the vegetative growth of the plant. Now, this SPL is under the regulation of a microRNA called MIR156. Temporally now, the levels of 156 go down as the plants mature. Since microRNA is a negative regulator, so that it is obvious that as the microRNA goes down, the level of the SPL will goes up and the vegetative phases will be regulated. Now, SPL is a transcription factor which regulates the expression of another microRNA called MIR172, which targets AP2. And AP2 is a transcription factor that acts as a suppressor for flowering. So as the level of MIR172 increased, it causes an increase in the level of MIR172. And once MIR172 starts increasing, the level of AP2 starts coming down. And at the threshold conditions, the plant will then transition from the vegetative stage to the flowering stage, and the flower formation will start occurring as the levels of the flowering suppressor come down. And these, now this is not as simple as what has been portrayed here, because these, at least these 4 different molecules, if not more, are under tight regulation of the environmental as well as the hormonal cues. And just to show another, cartoon here, to show how the different hormonal pathways cross-talk each other and they converge on different microRNAs to regulate the downstream response. So here is 172, which is regulated both in response to auxin and gibberellin and indirectly by ADA. And this is the one which is acting on AP2 to regulate flowering. So the signals in the plants are well perceived, and they converge in highly stringent or highly intricate pathways onto these genetic nodes, which are a microRNA transcription co-regulated nodes, to regulate thousands of downstream genes to bring about a particular response. And what happens if you try to change one of them? So this is a cartoon showing MIR156 overexpression phenotype. So this is what a normal Arabidopsis plant looks like. And if you try to overexpress MIR156, then the plant will become more bushy, and will a lot of leaves will be formed, and it will retain in the vegetative state. Similarly, tweaking a single amino acid can drastically change the phenotype of the plant, indicating that these are very, very powerful molecules. So how were the microRNAs, or how did the study of the microRNA progress in plants? Initially, it started with the genetic approach, which was the classical approach of identifying the genetic element which has the biological functions by creating mutants or genetic strains. This was followed by the standard biochemical approach of isolating the small RNA populations, cloning them meticulously, sequencing, and then characterizing them. And then came along with it started the computational approach. Initially, the database used were the ESTs, or the partial transcriptome or genomic sequences that were available. And these were used to identify the microRNAs. And then once the sequences were identified, it was easier to clone them and characterize them. So microRNA gene discovery by cloning, has been really supplemented by the computational approaches. And the first report of plant microRNAs in plants appeared as early as 2000, where a lot of different microRNAs were identified in the plant Arabidopsis. Our group also started contributing in this field, and by 2009, we came up by our first publication, in which we had cloned and validated the novel microRNAs from using the Pusa basmati rice variety, and at least 14, we identified 40 novel microRNAs, and at least 14 of these sequences were deposited in Mirbase by our group. And during this, the screening and identification, we not only identified the microRNAs, but also correlated their roots, their roles to different developmental organs by identifying molecules which show preference in shoots or flowers or leaves, or they act as crosstalk nodes to diverse responses like salt stress and virus infection. Okay. So coming back to the computational biology, which is the topic of today's talk, the computational biology really came up because microRNAs had well-defined structures, and the first part, that is the biogenesis, occurred in a structure-dependent and a sequence-independent manner. This is a classical structure of the precursor. And based on the study of different structures, specific patterns were observed. Like there is a hairpin loop, and there is a stem region, and towards the two ends, and the hairpin loop occurs in the middle. And then the cleavage would occur near to remove this loop as well as the overhangs here. And it is the microRNA would be coming from the stem region. In this case, there was perfect complementarity was not seen in case of plant microRNAs. There were some unpaired bases giving rise to structural features like bulges or mismatches. And but the size of the stem of a mature duplex remained around 22. That is between 20 to 24 nucleotide. And also, is there a question? So, okay. So, and also the free energy, since, this is a structure, folded structure, so it needs to be stable. So the free energy calculations were done, and it was found that a precursor should have a low free energy of or less than 30 calories per mole. Not more than 5 mismatches were tolerated in the stem regions. And interestingly, GU pairing was allowed. So based on these features, specific scripts could be written by the people, by the computational biologists or the bioinformaticians, and a lot of predictions were done used from the EST database. So much so that from the in the first version of MIG-base, there were about 500 sequences belonging to 6 species. And as these computational scripts started running and more predictions started coming, then slowly the number of microRNAs started increasing. And now we have the 21st version, where about 35,000 sequences are present from 223 different species. So not only the numbers of the microRNAs increased in the MIG-base, there are still maybe many more which have not been deposited, but this also got reflected in the terms of publications and other reports which were published by different groups working across the world. So this was how the computational biology provided a speed or an exponential increase into the discovery of the roles of microRNAs. Now, initial cloning and isolation followed by sequencing was a laborious process. And with the development of next generation or deep sequencing techniques, a brute force approach came into being, because now a lot of these sequences on a large scale sequencing can be done, and a lot of microRNAs could be identified. And with the development of the sequencing technique, especially a lot of inputs coming from Illumina, because it was good at sequencing the small RNA stretches. A lot of small RNA libraries were sequenced initially from Arabidopsis and rice, which were model plants, and now from a variety of different plant species. And a lot of structures were formed and predicted. And as the database increased, so this helped in refining the rules. So the structure still remained the same, but newer features were added to the rules, like the miRNA, and both miRNA and miRNA star sequence are derived from the opposite stems of the duplex, and the presence of both is mandatory in the library, at least in one of the libraries. And also the mature duplex should have a 2-nucleotide 3' overhangs. Then the nature of the asymmetric bulges was studied in detail, and it was found that the minimal base size of 1 or 2 bases was more preferred. And then a lot of comparisons were made between the structural feature at the, similarities in the structural features, because the first level of processing is sequence independent. So whether there are any particular structural features which favor the miRNA processing, and what kind of manipulations or mismatches were tolerated in these structures. For example, small RNA producing stem loops that are slightly violate any of the earlier criteria could still be annotated as microRNAs, but with the rider that these need to be experimentally validated later on to show that these structures actually exist, because it is very important and yet very difficult to distinguish between the real molecules from the system noise. So this really triggered the computational methods, the refining of them, creating more databases, as well as prediction algorithms. And now the web is full of them, and we have a lot of information coming in, especially from Arabidopsis followed by rice. For example, in stress, most of the literature was quickly revisited, and when it was known that stress regulates a particular response to, say, in response to drought or ABA, a certain set of genes like NFYA. And now you had the regulators, we could identify the microRNA regulators of these transcription factors. And with the help of NGS, their expression profiles were quickly captured. And it was known that when the stress comes, there are specific microRNAs which are downregulated so that the downstream transcription factors get upregulated to help the response of the plant be more the plants become more stress responsive. Or reverse patterns were seen that certain microRNAs were upregulated so that the susceptibility genes or the factors were downregulated. Ultimately, it is the permutation and combinations of these different genes which would regulate the overall response of the plant. So what our group did was we decided to work on rice, mainly because it's a model crop plant. And we looked at the different tissues over the developmental stages. We restricted our work to indica rice varieties as we wanted to build a database in the Indian context. And rice, as can be classified on the basis of the grain size into short grain, medium grain, and long grain. And it is the long grains basmati rice which is more preferred as compared to the short and the thick grain rice in India. However, the long grain varieties are more stress susceptible, relatively more stress susceptible as compared to these the short grain rice varieties. So we took varieties belonging to each size category, and the ones for which contrasting stress response was known. We also included a lot of transgenics and RILs in our data sets and studied these tissues in response, not only in normal environments, but in response to different stresses like salt, drought, high temperature, or virus infection, given individually or in combination, so that distinct data sets could be built up. The sequencing was done at the level of small RNA, at the level of the whole transcriptome, and we even did a specific degradome sequencing. Okay, so degradome, for people who don't know, is a specific kind of sequencing in which the transcripts that are cleaved by microRNAs are sequenced. This is sequencing of the cleaved transcripts. So we can note from this data set, when compared with the transcriptome and the small RNA expression profiles, we'll give an idea of which are the transcripts that are actually cleaved by the small RNAs, because there may be many more which undergo translation, are not cleaved and undergo translation repression. So a lot of this data was analyzed back to the genomes, a lot of one-way, two-way, and multi-way combinations and comparisons were done. And again, the major challenge for us was to again distinguish the system from the background noise. So we resorted to quick validations on a global scale using the microRNA, both for the mRNAs as well as the miRNAs. This threw up a lot of complex heat maps, but the ones which were showing similar trends on both the data sets were picked up for further validations. And based on that, we identified not only the known sequences which are present in the mirbase, but almost 2,000 novel microRNA sequences. Each of these were meticulously validated by PCRs and by northern blots, and their expression profiles were validated in the different tissues of rice. And the red, so these are some of the structures. The red lines, if you can see, would be the lines representing the MIR, and on the opposite strand would be present the MIR star sequences. So since this was done, relatively earlier in the time scale, so a lot of work was done in picking up each sequences, the sequencing sequence, analyzing its structure, and then carefully validating it. After PCR, the individual bands were cloned and sequenced to confirm that we are dealing with the same MIR sequence molecule, and there no variation exists in the sequence. So, and once the sequence was confirmed, then we could get back to the NGS datasets and retrieve their in silico expression profiles and build complex graphs which could be compared in tissue specific or stress specific or variety specific manner. We also got an idea of how the nucleotide composition of these microRNAs varied the length variations, and so on and so forth. So this was a lot of data which was sifted in many different ways and analyzed, and we could capture the expression status of 740 known microRNAs and 2,000 novel microRNAs, and almost all of them were wet lab validated. And interestingly, we had profiles of large number of these microRNAs which had no known role in plant stress responses, throwing up a lot of interesting and new information. So this is just another complicated slide, but you walk with me and I'll show you these are the northern blots to show the specific microRNAs which are coming up, which is present only in the leaf tissue, but is not seen in the root and the flower tissues. And another one which is coming up in the root, the level is too less in the leaf and is not there in the flower. So this gave us an idea of microRNA family members, which could be leaf specific, root specific, or flower specific. Then we went and then looked at how the target expressions were changing and whether they were showing any opposite expression pattern or not. Wherever the expression pattern was found, those microRNA target pairs were picked up. And again, we took help from the computational scripts, going back and doing a lot of network analysis to build the or to identify the genetic nodes which are acting in the tissue specific, stress specific, as well as variety specific manner. So this is just an example of how the map would look like in one plant. So this is a cartoon of a pokali plant. Pokali is a variety which grows naturally in the southern part of India, and it grows in the backwaters. So the plant is naturally adapted to tolerate salt. It grows in saline water. So this is the root and the shoot phenotypes, and based on this, we could build maps of how particular miRNAs are either going up or going down, and the behavior of the targets, and then what kind of plant physiology they would be regulating, and how they would be how the regulations in the root would be different than that from the shoot or in the flower morphology. So to talk a little bit more about this, what we did, we took the salt tolerant, that is pokali, and we compared it with the salt susceptible, that is the long grain Pusa basmati variety. Now, although these are contrasting salt behaviors, but they are two genetically different background, one being short grained and other being the long grain. So in this, we added the transgenics. These are the salt-tolerant transgenics developed by Professor Sapori's group by overexpressing the glyxylase pathway. And the background here is Pusa basmati. So we have the Pusa basmati susceptible as well as the salt-tolerant lines. And then we tried to make some comparisons with the naturally salt-tolerant pokali lines. And in this, we added one more set of transgenics which were raised in our lab. And this is these transgenics were a different biochemical pathway in which protein B2 is overexpressed. And this protein, this is a viral protein which imparts a salt tolerance to the plants. I'm not getting into the details of this, but so we had two different transgenics in the susceptible background, which were showing varying degree of tolerance to stress. And we had a naturally salt tolerant variety. So we compared them at the level of the MIRs and the mRNAs. Now, if we compare, say, the glyxylase plants with the Pusa basmati plants growing under normal conditions, all the red dots that you see, and similarly, the ones which are the two sets of plants which are growing under the salt stress conditions, all the gray dots that you can see, you see a lot of, there is a congested pattern and no clear cut pattern is emerging. There are a lot of there's a lot of variance with some, though some are getting upregulated, some are getting downregulated, and there's no clear-cut pattern or selected molecules that can be picked up. So we decided to look at this data in another manner when we compared the glyxylase transgenics, salt stress compared with the normal background, and similarly the Pusa basmati salt stress with the normal background. And now you have the graphs here, and these are much more better or much more cleaner to interpret as compared to the other graphs. And we have clear-cut idea of the molecules or the dots represented by individual dots that are getting down-regulated or up-regulated. And if we try to interpret this in terms of the charts here, so this is the baseline or the zero. So you see in the susceptible variety, there is a greater degree of down-regulation as compared to a greater degree of up-regulation in the tolerant variety. Now, what happens in glycolase overexpressing transgenics? Again, the pattern shifts with a greater number of upregulations as compared to the downregulations. The individual color bars represent the fold deregulation, twofold or fourfold or more. So let us not get into the individual color variations, because obviously this is only one biochemical pathway altered and a plant moving towards stress. And what happens in B2, which is another biochemical pathway, since these are slightly better tolerant to stress, but not very high tolerance level, the patterns here again are more towards upregulation there. That means the microRNA expression patterns are changing in response to stress. And this gave us the encouragement that now we can really fish out or zero down on molecules which may act as master or critical regulatory nodes to regulate the overall physiology of the plant. So we selected the molecules and started doing their profiles of individual molecules in different tissues like focally, grown under normal conditions. So there were genes which were getting microRNAs which were getting upregulated or downregulated. Similarly, in Pusa Basmati, made a comparison and found molecules which were showing similar patterns in the two varieties, and then plotted a threshold and tried to plot a threshold value to identify the ones which were going beyond threshold, like the bar here or here that you see. And these were the ones that we picked up for further functional analysis. So before I go into that, what we did was, so this is a list of some of the molecules which were coming up in showing a beyond threshold deregulation. Their targets were identified and categorized, and their expression patterns were, as you see in the heat map, they were opposite to the microRNA expression patterns, indicating that there was some kind of regulation which was a regulatory circuit or a regulatory group that was operating. And if we look at the characterization of the targets, we find here the channel transporters, we have the enzymes, and as well as the transcription factors. So then we took these and then tried to look at how they are working in the roots, because these are the tissues which are directly exposed to the salt vis-a-vis the roots, which get the signal of the presence of salt from the roots, and try to see how these the microRNA target nodes are behaving and how the expression profiles are changing. So what we found that the molecules which are upregulated in the roots in presence of salt are actually the ones which are downregulated in the roots in the absence of salt. So the presence and the absence of salt reverses the values. So these charts may not be very clear, but the paper is now published in JPGR, and you may go back and refer to the graphs here. And then since since this was mathematical calculations and we had the figures, we tried to plot it on the four sectors, trying to see the upregulations and downregulations. And this actually helped us identify molecules which were getting upregulated under all conditions, under salt stress conditions in both the plants, or they're downregulated in the plants under in presence of salt. And there were others which were showing different variations. So based on this and based on a lot of KEGG and geo analysis was done, and based on this, we could identify some of the key transcription factors which are regulated by these microRNAs. There were transporters like the SOS, the sodium potassium transporters, which are involved in probably regulating the signaling pathways. And these two pathways converge on the ROS, or the reactive oxygen signaling pathways, and some kind of correlation emerge. And when we looked at how these correlations are affecting the metabolic pathway, then we found that the key metabolic pathway regulating the respiratory pathways were changed. There were some kinases like serine, threonine, tryptophan, as well as the inherent glycolase pathway itself was getting affected in these plants. So all this, we're having all this data from different, at least 7 different rice varieties, and a lot of this data was validated. So we came up with a web resource which is freely available on the ICGEB website, and this is termed as ARMOR, for a rice miRNA mRNA interaction resource. And this can be searched and sifted or in different ways to get the required information, not only for rice, a lot of this, some part of this which is we have already published, but also for other plants like the sister plants, those of the grass family, sorghum, sugarcane, so on and so forth, because most of these MIRs are conserved. Their homologues can be found in these species, and a good method to find the crosstalk nodes and then pick up molecules for functional analysis to identify the loci that can then be suitable for genome editing or for mutational breeding or any other kind of downstream strategies to improve the crop response to tolerance. Just to show how the data can be utilized across different crop species, here we show the profiles of MIR 164 and NAC, which was conserved in tobacco, in rice and soybean. So we picked up from rice, and these are the actual qPCR plots to show how this particular MIR node is varying in rice, in tobacco, and between 2 soybean varieties, one tolerant and one resistant. And these, without getting to the details, even if you try to look at the overview, you can see a lot of variations in the way the bars are coming up in each of these varieties. So it is highly likely that the natural regulations of these microRNAs in the plants have gone a long way in helping the plant adapt to the niches in which they are growing and respond to the stresses. So the plants do have a mechanism to adapt, but over the period of evolution, this is a process which occurs very slowly. But these are the molecules which can be picked up and tinkered with to get to change the response of the plant quickly. So ultimately, I would conclude by saying that sequencing projects along with the biological computational are 2 powerful tools for studying gene function, and they can go a long way in resolving the complexity and dynamics of crop genes. Because there is much which is written in the genome of a plant, say rice, not only at the level of the DNA but also at the level of RNA. We have to look at how the genes are coded, including the splicing and the alternate splicing and the other events, how the non-coding RNAs are coded, how the two interacts, then how these regulatory nodes are regulated spatially and temporally, as well as over environmental niches to help the plant grow and thrive. So with this, I'll conclude, but I need to thank my organization as well as the entire team of members, those which have now left the lab and those who are still there in the lab, because it is their hard work and their results that I am speaking in front of you today. And of course, thanks to all the collaborators, because all this work could not have been done single-handedly, and to the funding agencies for supporting this kind of work. Thank you, and I'll be happy to take questions.
Thank you, ma'am, for this talk. Without delaying any other comment, I would like to invite Dr. Rajkumar for initial comment.
Very nice talk, Dr. Mishra. Um, I learned so much of the things, and we are also at KSAS doing some work with micro-RNA here
Mhm.
for treating, for developing a treatment for a spinal cord injury. Uh, so my question is one of the question I have that you showed in one of the slide that the microRNA they are very conserved and their regulation is very conserved in that sense. So they also getting affected by the uh, environmental changes if they are their regulation is conserved.
Yes, microRNA expression profiles change in response to stress because the expression microRNAs arise from endogenous genes. They are coded by the same RNA polymerase 2 promoters which code for the other transcripts. Okay, so they have the same kind of cis elements like the ABRE binding elements or the transcription factor binding sites. So their expression is very much regulated in response to environmental stresses, hormonal responses, so on and so forth.
And
Also, this
epigenetic
is,
changes,
I am coming to that. Also, the fact that microRNAs are conserved between plants and animals. This is also correct. But between plants and animals, there are subtle differences in the microRNA pathway. I spoke only with respect to the plants in my talk. What I would like to add now here is, like I showed, that the entire biogenesis of the microRNA is occurring in the nucleus in the plant nucleus. Okay, but in an animal nucleus, the only the precursor is produced. That is the primary first step and the second step. The primary and the precursor molecules are produced in the nucleus. The precursor is then sent to the cytoplasm, where the mature microRNA gets diced out and then gets incorporated into the RIS complex. Similarly, at the functional level also, the stringency is much more tighter in case of plants as compared to animals. In plants, we talk about the whole sequence matching. But when we look at animals, we talk about seed sequence matching. Okay. And this is understandable because plants are sessile and animals are motile. So there are some differences. Having said that, both RNA polymerase 2 is the one which is giving rise to the miRNAs, whether it is plants or animals. So you can expect the same kind of regulations in your animal tissues as well.
Yeah, just to add, is there any epigenetic changes which is also regulating this?
Absolutely. So microRNAs are maybe I should have had a basic lecture on microRNAs. MicroRNAs come in 4 size classes: 21 nucleotide, 22 nucleotide, 23 nucleotide, and 24 nucleotide. 21 nucleotide are the canonical microRNAs, which are involved in the PTGS pathways. 24 nucleotide microRNAs is the size class which is involved in TGS, that is transcriptional gene silencing, by bringing about changes at the epigenetic level. And there they interact with a lot of these chromatin machinery, mediators, and the other complexes to regulate whether the gene expression can be switched on or switched off.
Okay, thank you.
We have Professor Balram Singh sir here. So, sir,
Maybe, to Dr. Rajkumar's comment, I should add one more point. NGS is the same, whether it is the plant tissues or it is the animal tissues. Ultimately, it comes down to sequences. The kind of scripts or the kinds of logics that you would use when you are looking at trying to identify the plant pathways or the animal pathways, there will be slight differences. Okay, so the most common mistake people do is take animal sequences and then run plant softwares on them, because a lot of softwares are freely available also online. So that care should be taken. The logic for animal analyzing the animal microRNAs and the logic for analyzing the plant microRNAs will be slightly different. And those differences should be carefully, incorporated into the algorithms to get the right kind of results. Otherwise, this differentiating system from the noise will become very difficult.
Okay. You are muted.
Oh, sorry. We have Professor Balram Singh, sir, the Director of Institute of Advanced Sciences here. So I would like to invite you, sir, for comment.
Yeah, my video is not working. I'm so sorry.
No problem, sir.
Professor Mishra, it was a very nice presentation, very comprehensive. I missed half of it because I had another meeting. So I'm so sorry about that. But I was looking at the slides while I was talking. My school has a meeting. There's a school in my village. On Wednesdays, we talk to the principal for half an hour. So in the middle, I had to go there. But it looks like you have a very large team of people working for a long time. And microRNA, you are now addressing microRNA not just a technique, but really comprehensively to understand how microRNA has a major role. What, from my point of view and Rajkumar's point of view, is even in the process of evolution. So you're talking about plants, you're talking about animals, and microRNA is becoming a constant thing. So what do you think in terms of evolution? Is there microRNA is just an instrument of evolution or is being also evolved?
Well, Ramji, सबसे पहले नमस्कार. Because you are a senior and also as a director and also as a as the organizer of this symposium. So my greetings to you. I don't have a very big team. We are about 7 people in the lab at one point in time. But yes, since we have been working on microRNAs for a long time, so I have a lot of people who have superannuated or have graduated and left, and I make it a point to include their name because we are running a relay race. What the first person started, the second person has taken the baton and we are running, so we can't leave the first person out. We are probably the downstream generations are now in the lab, but the baton is being carried forth. So that is why you see a lot of names on the last slide. Now coming to microRNAs, yes, microRNAs is a passion for me. Within the non-coding family, microRNAs has always fascinated me because they are, one, they are endogenous, two, they are conserved, three, although they are conserved, there are functional differences between plants and animals, right? Nature will not evolve something which is not useful. I go by this, this understanding. If something has evolved in nature, then it is useful. It has competed out, and that is why it is there. So this shows that microRNAs are very, very important. They are regulatory molecules, and that is why they have involved. During evolution, differences has come up between the animal lineage and the plant lineage because of their inherent, properties or requirements of the two systems. So this indicates evolution. So evolution has definitely happened. They are evolving species. And even now, when we look at molecules, we find some of them, the targets are no longer there in specific plants or in specific tissues. This means they are still undergoing evolution. There are newer molecules or molecules with, SNPs or single nucleotide changes which are coming up. And there are some which are losing their targets. There are a lot of these microRNAs which are also evolving from the retrotransposons. And they are the most interesting molecules because although on one hand, their function is to keep the retrotransposons silent, but on the other hand, they allow these retrotransposons to become functional at a point in time, and making a very generic statement, at a point in time, so that it can help the organism adapt to the changing environment or the niche. And especially rice, we see a lot of varieties. Even if we look at our country, we have rice which is growing in the backwaters. We have rice growing as a lowland rice, as an upland rice, even a high altitude rice, which we find in Kashmir and the other areas. So these plants are very different. You bring one rice variety from Kashmir and immediately plant it in Kerala, it will probably not grow. But over the period of time, the plant may adapt. So the plant has the mechanism, it has the information, and with both the microRNAs as well as the normal transcripts, they are co-evolving to regulate the response of the plant. And this flexibility is required because the moment the flexibility goes away, the homeostasis becomes rigid, and this would impact the evolution. This is my understanding.
Okay, very nice. I hope. Well, let me ask you one more question. Is there? I understand what you're talking about, plant and animals. Is there within animals? Is there gradation in miRNA within just animals?
Yes,
or
there
plants,
is.
or the plants also? The lower plants and higher plants, do they make different kind of microRNA use or different pathways?
Yes, there is a gradation. The conservation of plant microRNAs is seen within the plants. The plant homologues are, a large number of plant homologues are absent from the animals and vice versa. But of late, I would say in the last 2 to 3 years, reports have started coming in by select groups of people who are working. And this has also triggered a lot of controversy, where they are saying that some of the plant microRNAs have been found in human blood and human tissues, probably coming from the food that we eat. Now, if they are circulating in the hypothesis which the authors have proposed, and this is, I'm talking about the published paper, that they say that since they are there in the human tissues, they have a function and they are probably regulating some of the human tissues. And this information has triggered a lot of controversy and also a lot of excitement in trying to identify if there is a crosstalk. And there is a stream of research or rather scientists who are interested in looking at and trying to understand, one, whether the crosstalk exists, and if so, how it happens. The crosstalk can be plant-plant, it could be plant-animal, it could be plant-fungi, it could be in many different permutations and combinations. But this is one area of research which is coming up, and there are a few papers which are already existing in nature, in literature on that.
What I may have missed this in the introduction. What is the origin of miRNA, microRNA?
Okay, so microRNAs are coded by the genome. If we go by our classical or old knowledge, within the chromosome, there was a large part which was considered as a junk.
Yeah.
There was a coding region and there was a region which was labeled as junk. Now we know that, because whatever we don't understand, that is human, we categorize it as junk because we don't understand. But this junk region is coding for transcripts which do not make proteins, but they make these small molecules, small RNA molecules, which have regulatory functions. There are microRNAs, there are TACs, there are different kinds of these molecules. One subclass is the microRNA, and these microRNAs have a regulatory function. So the idea or the emerging idea or the changed concept now is, now there is nothing like gene on and gene off. I mean, protein formed and protein not formed. But rather, we talk about gradients of protein. As these gradients, and this gradient is established due to a very delicate balance between expression and silencing. And this is what the microRNA and transcription factor interactions are doing. And we are
Thank you.
far from understanding them.
Thank you. Thank you very much. I
Uh,
was just speculating maybe this RNA may have initial origin from virus which got integrated in chromosomes.
There are viruses which bring microRNAs. These are called viral-derived microRNAs, and they have a role in infection. That is a different line. But every organism codes for a set of microRNAs which come from their own genome, and they do different functions. The microRNAs are responsible why a rice leaf will look different from a maize leaf, although both are grass family.
Okay. Thank you. Thank you so much.
So one thing I learned from today's presentation is that if you think that the upregulation of the gene, if you can achieve that and try to see a desired result, but you are seeing something very different result in case of the coloring of the flower. So that's kind of a telling very interesting thing to me, that at least that we are trying to develop some genetic medicines, and we are trying to see whether we can upregulate or downregulate some genes in the body on the inside the cells. But we are also plant is giving that signal that that achievement is little bit tricky for getting the desired result. And also if you um, uh, refer to the Dr. Is a book, Selfish Gene. Uh, I do, Dr. Singh, do you remember who's one of the
Yeah,
authors? About Richard Dawkins, right?
Dawkins, yeah.
Richard Dawkins. So in that also he said that the genetic regulations is not mean to be solely for the protein production. So he
Absolutely.
mentioned, yeah. So he's mentioned
Absolutely
that.
right.
So
See,
the genetic
this is where
regulations being something more than what we know actually.
this is where the regulatory RNAs come into play. See, a genetic makeup, your genetic makeup and my genetic makeup, since we are classified under humans, if we go by the genome, is a human genome, right? But now we are getting into individual genome sequencing, personalized medicine, personalizing, because these are variations. And these variations could be at any level. It could even be at the level of how the microRNAs are interacting with the genes. And since you talked about medicine, I'll briefly mention that we have very recently published a review in phytotherapy, where we are showing a concept. We brought out the concept. You know, many plants are used as phytomedicines. Phytomedicine, you know, plants as medicine. So when the concept was that when you are taking these decoctions, which are plant-based decoctions, they are rich in antioxidants and metabolites, which are going in and doing something to our genetic architecture, and therefore it is boosting our health or maybe suppressing cancer or many other things. What now research is showing that when we are taking these decoctions, along with the proteins and the metabolites, we are also consuming the microRNAs. And these microRNAs may have a function in regulating the host genes. And in that paper, we have cited published literature to show how microRNAs can regulate the phytometabolite levels in the plants, and the phytometabolites, when consumed, go and regulate the microRNA levels in the humans, and this would affect the health state of the human, whether a particular cell can be healthy or can be diseased or cancerous or whatever. So this again is a published article, and this, if this cross kingdom regulation or crosstalk actually exists, and we are able to validate some of them, this is going to really throw up a new new view on the whole system, where the basic concept is food is health, right? So the science behind food is health would change if this cross-kingdom regulation is actually proved. This is still controversial, so I am being a little more skeptical about the whole, the whole thing. But there is potential there, and especially with respect to
One more thing.
traditional Indian.
Yeah,
Sorry, I tend to speak more because especially with respect to traditional Indian knowledge, because there is a vast literature there, because traditionally we always consumed balanced diets, right? And
True.
that will have lot more implications in understanding this if these cross-kingdom regulations can be understood. Lot of correlations which the present day scientists
Yeah,
say.
we call it junk. What we don't know, we call it a junk gene, but we tend to eat junk food. And also, I just want to add one more thing is into this discussion that the COVID-19 vaccine developed by Pfizer and AstraZeneca, they are a genetic medicine, and then more and more side effect we are seeing because of what we are discussing here. And probably that may be one of the reason why Harvard once had a genetic institute, but they closed that genetic institute because they probably think that the genetic medicine are not going to be they may be a tricky thing for development.
Yeah, quite possible. See, ultimately, what we know standalone, especially say in case of cancers, there is lot of literature. You are aware that micro-RNAs get deregulated in response to cancers or in terms of other neurological diseases like Alzheimer's. This is published literature. People have shown that there are deregulations, right? Then we know that there are molecules which can modulate the micro-RNAs. These molecules can be phytomolecules, these can be any other kind of chemicals, these could be synthetic molecules, anything. So it boils down to that what you need is regulators to control which way you have to go, but you have to understand the regulation before using the regulators. And that is what we are trying to understand. But we are doing in plants, and there are other groups who are working on the mammal systems.
Okay, thank you very much.
Yeah, all right. Thank you so much. Thanks
Yeah,
for
please.
this
One
Yeah.
question. There is one question from
Yes.
Abhinandan.
My name is Abhinandan, ma'am. I'm also scientist in KSAS. I learned a lot from you and Dr. Shapuri sir because I did my PhD from NBRI Lucknow, and my PhD topic is also related to micro-RNA. And I work on a Rhodopsis micro-RNA, and actually I collected the different Rhodopsis plant from different altitude of Himalaya and trying to find the role of micro-RNA in the adaptation at different altitude. So when I analyze the microRNA, I find some microRNA showing low expression. Some are as expected, some are higher expression. But when I choose one microRNA and compare the what is called the precursor, not primary transcript and then precursor. When compare the precursor and primary transcript, I find some deletions, and that's why that microRNA show down regulation. So you have multiple rice varieties, and you also got some up-regulation and down-regulations in your microRNA. So did you try to check that? Do you have any insertion and deletion in precursor?
Absolutely. Because I told the RNA polymerase II mechanism, which makes a normal transcript or an mRNA in the cell, the same mechanism holds for the primary microRNA transcript. And the primary microRNA transcript also undergoes splicing and other events. Editing and splicing happens, and that is probably where the changes happen. Okay. Not only that, microRNAs also undergo post-processing changes. Again, we have published this thing where we show that the ends either at the 3' end or the 5' end, some nucleotides will change. This can happen in a template dependent manner. That means where the DNA template, the similar sequences are added, or it is done in a template independent manner. That means arbitrarily, I am saying arbitrarily, or due to certain unknown phenomena, some sequences get added. When this is happening at the 5' end of the microRNA, even if you change 2 nucleotides, it will change the targets of the microRNA. Okay, because targeting is sequence dependent. The moment 2 nucleotides get added, the entire sequence changes. Also, what many people have not looked at, there are dicers and there are AGOs, the two main molecules, microprocessor and the dicing complex. In a plant like rice, there are 8 dicers and there are 10 AGOs. Okay, maybe more, because there are isoforms also. Now the system, this is probably evolved for redundancy, but yet the system would have which AGO will take care of which microRNA. We have 4 forms, 21, 22, 23, 22. Yeah. Okay. We have about 9 AGOs. Then we have 4 bases, A, G, U, C, T. So now 4 size classes with the possibility of starting with any of the 4 sequences. And then you have a diversity of AGOs. So which molecule picks what and what is the role? Whether this particular AGO is taking care of splicing, or it is taking care of translation repression, or it is getting into the TGS pathway, we don't know. So the variations are there. So there are SNPs, there are deletions, there are sequence variations, then downstream target also variations. So there is a huge complexity. First microRNA in plant was identified in the year 2002 from Sunker's, Zhu's group, Sunker and Zhu first paper came. We are in 2025. We have a vast amount of literature which needs to be revisited to understand what is happening. Right. And so you did a very interesting piece of work. Try to work more on it and try to give us an information of what is happening. Whether these SNPs are actually, or these changes in sequences are actually helping the plant adapt or not.
Yes, of course, I identified and find that particular, actually I'm first time validated one microRNA, I think you are aware of that. But that is only found in apparatus, microRNA 158. And first time I validated, that this microRNA helped in producing Phagy RNA.
Phases.
Phases. Yeah, tRNA or Phagy RNA, tRNA. And that microRNA size is 20 nucleotides, majorly expressed 20 nucleotides. When microRNA is 22 nucleotide, and some microRNA are in size of 21 nucleotide, and target specific position, they start producing tRNA. But I find that when the size of a microRNA is 20 nucleotide, then this microRNA negatively regulate the production of tRNA. That is the interesting finding of that study. And in addition to that, ma'am, I also did my postdoc in Israel, as we discussed about the conservation of microRNA pathway between plants and animals. And that group is working on the cnidarians. And cnidarians, is a very basal animals. And cnidarians, the target cleavage and mRNA production is not like animals; it's like plant.
So that is the evolutionary transform probably. Yes, we have a long to go and to understand what is happening there. See, when we take because we have a lot of NGS data, when we sift through the NGS data, these are sequences, right? You can compare it with plant genome because we know it is coming from plant. So we just decided to take these and compare it with the human genome. And we found at least 1% of human microRNA sequences were matching. Now, how do we interpret it? Do we say that human genomes, human microRNAs are present in plants? Or do we say that, there are sequence conservations? This can said only by experimental validations. But computationally, many of these permutations and combinations will come up. The interpretations have to be made very safely after a lot of experimental validations.
Thank you, ma'am.
So, thanking you, ma'am, for joining along with all who have joined this KSAS talk. And as the directors of KSAS and INADS mentioned that this was very, explaining talk which you delivered here. And INADS have this kind of missions to explore, to provide the scientific discoveries and study the social science scientific discoveries in the society in layman style. So these kind of research will definitely enhance the social outcomes, as I mentioned earlier, that this will definitely help to improve the rice varieties by enhancing stress resistance, disease tolerance, and many more things as you mentioned in your talk. And this will also, help the farmers and will benefit them more resilience crop that requires, fewer pesticides. This is also the problem in the society in this time. And it would also help the consumer to get the access the healthier, more sustainable rice with improved nutritional values. So these kind of research are very much needed and must be done in the society by the scientists, and the social scientists have the responsibility to spread in the society as beneficial targets. So I am thanking you for joining us, and we will again invite you to listen the further progress for this project, about this project. Thank you.
Thank you.
Thank
so
you.
much.
Thank you
And
very much.
to all.
Thank you.